The use of C. elegans, a widely studied model organism, allowed the team to study the unique and complex interplay between genetics, diet, microbiota and other environmental factors that can affect fundamental metabolic processes in different individuals. The research, by Marian Walhout, Ph.D., the Maroun Semaan Chair in Biomedical Research and Chair and Professor of Systems Biology at UMass Chan Medical School and collaborators Erik Andersen, Ph.D., from Northwestern University and Frank Schroeder, Ph.D., from Cornell University, published in Nature, Iden. The strain, known as DL238, has an abnormal accumulation and secretion of the metabolite 3-hydroxypropionate, 3-HP. Moreover, this strain was found to generate a set of novel metabolites that have 3-HP conjugated to several amino acids. This work provides an important step toward the development of metabolic network models that capture individual-specific differences of metabolism and more closely represent the diversity that is found over entire species, said Walhout. Employing this system, we can begin studying inter-individual metabolism and the unique interplay of metabolites, diets and environments on an individual level. When the human genome was sequenced, clinical researchers envisioned an era when our personal genomic information could be used to tailor. Despite the completion of the Human Genome Project in 2003, and advancements in genomics and deep sequencing technologies, personalized medicine remains more promise than reality. Part of the challenge in developing personalized medicine is that our DNA makes up only a portion of human health. An individual's diet and environment both profoundly impact metabolic processes. And because no two individuals have the same exact diet, unraveling the complex interplay of genetics, diet and environment and connecting these to variations in metabolism is cumbersome. In addition to sequencing individual genomes, scientists would need to replicate metabolic measurements in people of the same age and gender, who ideally would also consume the same exact diet and experience identical environments. To address this challenge, Walhout, a leader in metabolism and gene expression research, teamed up with Dr. Andersen, an expert in quantitative genetics, and Dr. Schroeder, a chemist, to develop a comparative system for studying inter-individual variations in metabolism. The group designed a system where environmental conditions and diet were constant among individuals with variable genomes, much like our genomes vary from person to person. We collected about 100,000 animals from each strain, and because they are all grown in the same conditions, given the same diet and have the same genome, it's possible to explore how genetic differences among the 4 strains impact metabolism. It's like comparing 4 different people. At its core, metabolism is the set of life-sustaining chemical reactions in organisms. The 3 main purposes of metabolism are the conversion of food into energy for cellular processes, the conversion of food to building blocks for proteins, such as lipids, nucleic acids and some carbohydrates, and the elimination of wastes generated by these 2 processes. A series of experiments including gas chromatography mass spectrometry, high-performance liquid chromatography mass spectrometry, and metabolic network analysis were performed and analyzed to identify possible differences and variations in metabolites between the 4 strains. As a result, more than 20,000 likely metabolites, the small molecules that collectively carry out metabolism, were detected, most of which remain unknown. When researchers compared the presence of metabolites between the 4 strains, they found more than 200 metabolites that were highly specific to one of the strains. These studies showed that 3-HP is formed during propionate breakdown via a B12-independent metabolic route, or shunt. 3-HP is then metabolized by the HPHD1 enzyme and ultimately converted into acetyl-CoA. In the current study, researchers were able to trace the abundance of 3-HP molecules in the DL238 strain to a variation in the HPHD1 gene, which allows 3-HP to accumulate. To compensate for the excess 3-HP, the DL238C elegans developed a mechanism for shunting the excess molecule out of the animal cells by cojoining 3-HP with amino acids. "We're just starting to scratch the surface," said Walhout. "Our study only uses 4 strains, but the next step is to see what we find when we look at 100 different strains. Or what happens when we use the same strain but vary the diets. We've put together a really robust model for measuring metabolic variation between individuals," said Walhout. "What made this possible, more than anything, is our unique, multidisciplinary collaboration. It's the expertise that each lab brought to this project that enabled this discovery. Bennett W. Fox, Olga Ponomarova, Yang K. Lee, Gaoshan Zhang, Gabrielle E. Gizi, Melissa Walker, Nicole M. Roberto, Huimin Na, Pedro R. Rodriguez, Brian J. Curtis, Aidan R. Kaladzij, Timothy A. Crombie, Stefan Z. Thanks for watching. Please subscribe my channel for more videos.